You can start, Eliese, please.
Okay.
Yes.
Okay.
Pode começar, por favor.
Well, good evening, everyone. It's my pleasure to say welcome to Professor Laufer and Serafin tonight. In fact, in Germany, the time is to drink a beer, not to work in a conference, but the jet lag obliges us to do something every time very, very new. And then I needed to enforce my thank you to both Serafin and Laufer for this attendance to here this night. Thank you to join us. Immediately, I invite Professor Laufer to come to this moment in our virtual floor. Professor Laufer, please.
So thanks a lot for this very kind introduction, and thanks a lot for giving me repeatedly the chance to bore you with my presentations. But please complain with the organizers, because all of them know me, and if I can't tell you too much new, then you know with whom you want to complain. Okay, so is the sound okay, and the picture, the presentation is okay? Yes?
It's perfect.
Yes.
Good. So then I try to summarize in less than half an hour our Tübingen's approach for academic drug discovery. And then as a case study, as an example, Ricardo will present his latest publication. So this is the main part, much more important than my introduction, but I want to give you a certain framework how academic drug discovery works in Tübingen. And we are very complementary to your INCT. And so I'm very proud that since a long time we have a strategic partnership of our drug discovery center and the INCT-INOFAR. Okay, so we are in fact a platform in the so-called excellence initiative of Tübingen University. You know, we have in Germany, like your print program, an initiative of excellence universities, and 11 out of 100 universities are so-called universities of excellence. And one of these is Tübingen. And we have two formats in this excellence initiative. We have the clusters and the platforms. And one of these platforms is the Tübingen Center for Academic Drug Discovery. So what is the background of such a center? The background is the situation that over the last decades, over the last 30 years, the number of new drugs which came to the market is more or less flat. We talk always about a number between 25 and 40, 45. You see here the light blue ones are the new molecular entities, the small molecules, and the dark blue ones are the new biological entities. So the light blue ones, we talk, as said, always something 30 to 40. And over the last years, we didn't have any more blockbuster drugs. We had just niche products. So it looks nice here with 42 in 2018, but half of them were niche products. And this puts pharmaceutical industry in a big problem. The problem comes from the fact that the spending of R&D stopped to increase more or less 10, 15 years ago. We had in the past always an increase of 10 to 12% in R&D spending, but as you see here, since about 2009, 2010, it's flat. It's still a high number, but there is no more increase. And this no more increase brings us to the point that the total costs for a new drug are currently $2.6 billion US. The last number is from 2018, and in 2018, one new single drug costed $2.6 billion US development costs. So what is the reason for this lack of increase? You know, in the past, we had always a good increase of more than 10% every year in R&D spending, but now, since more than 10 years, it's very flat. So the reason is that very that many of so-called blockbuster drugs went out of patent. They dropped down the so-called patent cliff. For example, Lipitor, atorvastatin, and so on. Many of them in the last 10 years lost patent protection. And this loss of patent protection brought big pharma, big pharmaceutical industry to a point that they are no longer in the position to spend a lot of money in R&D. And this is reflected as well in the so-called freshness index. The freshness index shows you how much of the sales of a company is coming from new drugs. And the greener it is, the more sales is coming from new drugs. And you see that only one company is more or less driven by new drugs, it's Gilead. All the other ones here are driven by sales from products more than 10 years old. And even the big three, Roche, Novartis, Pfizer, make only 10% of their sale from drugs younger than 5 years. So, and this really puts big pharmaceutical industry in a miserable position. So they have no longer cash cows to finance early research and development. And what did they do? They all stopped or reduced basic science, but teamed up with academic groups and with spin-offs. For example, AstraZeneca cuts 2,000 jobs in early research. Sanofi eliminated 900 jobs in early research. Merck & Co. teamed up with universities. Novartis did the same, Bristol Myers did the same, and Pfizer did the same. And as a consequence of this, 60% of the phase 1 projects, the one who enter first into man studies, is no longer coming from big pharma in-house pipeline. So we have now really the situation that big pharma is depending on drugs from biotech and from academia. And this citation here is from a business report of Ernst & Young. A huge, long, profitable industry, the big pharmaceutical industry, has been brought to a point of inevitable transformation. Inevitable transformation means they no longer fill their pipeline with own drugs. They need academia, they need biotech, and they need small startup. And this is now a big chance for academia, which we should take. And you take it with your INCT-INOFAR, and we take it with our Tübingen Center for Academic Drug Discovery. In my field, I am a kinase guy. We have currently 70 drugs on the market, and almost half of these 70 drugs are not coming from the in-house pipeline of big pharma, but are coming from academia. So even in our field, a very new field, a very rich field, a very big money field, big pharmaceutical industry is relying on collaborations, on biotech, on startups, and on academia. As a consequence of that, 15 years ago, the Academic Drug Discovery Consortium was founded in the US by Marcy Glicksman in Harvard and by Stephen Fry in UNC Chapel Hill. And currently, more than 100 centers of this Academic Drug Discovery Consortium are spread all over the world, most of them in the US. In Germany, we have 3 ones. And these 3 ones you will see later. One of them is Tübingen. And what is the idea behind these centers? The idea behind these centers is to help each other, not with confidential information, but with general information, with organizational stuff, just with consultancy, and to make, by such an approach, a big pharmaceutical research community all over academia. And as said, there are many members in the US, but only few outside. And in Germany, we have 3. And probably this can be one of our next approaches to bring some of the centers of Brazil also in this Academic Drug Discovery Consortium. This would be a big foster and a big booster of this scene in Brazil, I'm very sure. So where are the problems? The problems is that academia is very strong in exploratory research. We are good in target identification, but we are very, very weak in validation. You make an experiment normally once, and if it worked nicely, you make a publication, and you avoid to repeat it. Probably the second time, it would not give such a good result. Industry is exactly the opposite. They make confirmatory research, and this is heavily regulated by laws and by guidelines. And to bridge this gap between exploratory research and confirmatory research, you have to cross the so-called valley of death in drug development. In the past, we always thought the problem is here the lack of money, and we filled up this gap just with money, but this didn't solve the problem. So the real problem is really a clash of cultures. In industry, everything is regulated by law, by the ICH guidelines, by the Arzneimittelgesetz, by GMP regulations, and so on, by the EMA, by the FDA, and so on and so on. In academia, we have only one regulation. It's good scientific practice. And I promise all of you that nobody of you will stick to this good scientific practice. Even me is not doing so. So we have one single rule and we are not doing it. In industry, they have hundreds of rules and they fulfill all of them. And this is the real clash of cultures. Another clash of culture is that the rewarding systems in academia are totally different from industry. In academia, publications with high impact factor is most important. For what do you get high impact factor publications? You get for sexy papers. The sexy paper is new, spectacular, speculative. In industry, you have to keep your mouth shut. Everything is confidential. You work on projects with milestones. You have to file NDAs. And finally, you rely on the sales and the return of investment. This system is totally 100% different from academia. Then if you are important in academia, you have to be visible. You have to be a big ego. So like me here, whoa, whoa, I'm the best. In industry, you have to keep your mouth shut, keep everything confidential, make patents, do not publish anything. And again, this is the real valley of death. We have to switch, we have to bridge, and it's not only money. The classic approach in Germany, at least, was that academia is doing early target identification and late clinical studies because we have the big patient cohorts. But in between, the tendency of industry was always keep academia out. And medicinal chemistry for a long time was seen as an industrial discipline, but nothing really for academia. But this was a German problem. Outside Germany, in France, for example, Camille Wermuth, only 120 kilometers from Tübingen, he invented 4 drugs which are still on the market. In Austria, also not far from here, Dieter Binder, with his thiophen bioisostase, made 3 registered drugs. In Italy, Roberto Pelligiari from our partner university in Perugia founded a company in the US with a big market capitalization. And from Basel, also very close to Tübingen, Beat Ernst did the same. So it was everywhere possible, but not in Germany. So 10 years ago, we decided to do something different to fill up in probably 10 years from now, also 1 or 2 or 3 names on this slide here. As said, we started our initiative 2012, our Tübingen Center of Academic Drug Discovery, and we became in 2015 member of this Academic Drug Discovery Consortium. Currently in Germany, there are 3 members. It's the Pharma Zentrum in Bonn by Christa Müller. It's the European Screening Port in Hamburg, where another postdoc from our group did in part his PhD. He is also Brazilian. It's Thales Kronenberger. And the 3rd center is our Tübingen Center for Academic Drug Discovery, the TÜCAD. So why is Tübingen a good place? Tübingen is a good place to do so because we have a strong history in spin-offs. We have a good history in small molecule, new chemical entities, first into men, 4 molecules from our group. We have a phase 1 unit dedicated for first into men studies, and we have even GMP facilities for the API production and for the formulation of the trail medication. And we have complementary expertise with pharmacogenomic institution of the ICEFA, which is also part of our TÜCAD, and with the Robert Bosch Foundation in Stuttgart. So all of this together make Tübingen a good point to do such thing. So if you now want to do something new, you should carefully analyze what people need and what is already available. And if you look to the unmet needs in drug discovery and drug development, you have to check what is the reason for failures. And the reason for failures in the 90s was mainly this blue part of this chart, and this is pharmacokinetic. Why? In the 90s, animal experiments were replaced by cell cultures. But with a cell culture, you cannot make pharmacokinetics. In the 2000s, all of a sudden, toxicology, the yellow part, became the main attrition rate in drug development. Why? Because you wanted to reduce animal experiments and did everything again in cell cultures. But cell cultures are not replacing animal experiments, not in pharmacology and not in toxicology. In toxicology, even less. And what is now in the last 10 years, the most important attrition rate for the failure of new drugs, it's efficacy, lack of efficacy. And lack of efficacy, the green part here, is nothing else than lack of target validation. Lack of efficacy means that your 70-kilogram patient is not doing what your 30-gram mice predicts. And this is, in one sentence, it's badly done, target validation or some really misleading studies in target validation. So we decided to focus here on this green part, and target validation, as you know, was always important in academia. So we took advantage of the expertise academia has, target identification, and added to this target identification, target validation as an important step. The other important thing in early drug discovery is medicinal chemistry. And if I would have asked you where is the biggest cash trap in drug development, you would have mentioned phase 3, but this is not true. Lead optimization is more expensive than phase 3. Why? Because 95% of the money of lead optimization is for the garbage can. Because most of the compounds you synthesize here will never be useful for anything. So you sink a lot of money in the very early phase. And this is again a problem which academia can help. We are for sure not better here, but we are cheaper. A full-time equivalent in academia in synthesis is much less expensive and sometimes more creative than the people in industry. But what is important, you have to do real medicinal chemistry. What is real medicinal chemistry? Real medicinal chemistry is chemistry in which the properties of your compound are more important than the synthetic pathway to the compound. This sounds trivial, but it's not trivial. In the past, natural product chemistry was thought to be the holy grail of organic chemistry. And if you can synthesize complex natural products, you can invent drugs as well. But this is not the case. Why is this not the case? In a nice paper here by Dean Brown from, at that time, AstraZeneca, he investigated the 20 most important reactions in medicinal chemistry and the 20 most important reactions in natural product chemistry. And the overlap between the two fields are 2 reactions, 2, not more. So if you are a good natural product chemist, you are by definition not a good medicinal chemist, and vice versa. So this is sad truth, but it is the truth. If you want to make good medicinal chemistry, you have to think totally different from natural product chemistry. This is a newer study from Nature Reviews Drug Discovery, and this is important here, cost per launch capitalized. And you see here that lead optimization costs $414 million US, and phase 3 only 314. The capitalization makes the difference. So if you compare now these numbers, you clearly see that the biggest cash trap you lose the money during development is medicinal chemistry in the early phase in the lead optimization. So if you are good here, if you are cheap here, and if you are fast here, you can gain a lot of money and a lot of expertise in the field of drug discovery. Okay. So now something went a bit strange. Oop, I lost some of my slides, but don't worry. I can do it like that as well. Okay. In our Tübingen Center of Academic Drug Discovery, we focused on techniques which are called genetic optimization and genetic target identification and validation. And by doing so, we identified a pathway in liver cancer, and this is a resistance pathway. And by bridging this resistance pathway, we were able to come to a first-in-man study in a very, very short time. Short time means that the basic science we published in September 2014, and the first patient was treated 16 months later. And this was done in a fully academic setting. And this wouldn't have been possible in industry. In another project, we did the opposite. We published basic science as well, but we were not able to do it academically because we ran out of money in this project. So what we did here, we went to venture capital very early and took this venture capital for founding a company. And with this company, we finally made it now last November to first-in-man studies. So this took substantially longer, but this was fully financed by venture capital, and this was fully financed by academia. None of the two projects was financed by big pharma. So it was definitely possible to do it academically, to do it venture capital financed, and without the impact and the money of big pharma. So these were our two flagship projects, and all in total, we have 4 projects now, first-in-man, coming from our pipeline. Okay, but do we really need new small molecules? Do we need new drugs? Well, in my opinion, yes, and it can be easily proven. We have 3,000 potential targets for small molecules. And this is an offspring of the Human Genome Project. Out of this Human Genome Project, Hopkins made an analysis of the so-called druggable genome. And he defined the druggable genome as genes who encode a protein which has a binding site for small molecules. And if you take this definition, we have 3,000 potential targets for small molecules in our genome. As of today, we have 4,000 small molecule active pharmaceutical ingredients worldwide registered. So you can think, okay, we have 4,000 drugs on 3,000 targets. Everything is fine. No, it's not, because these 4,000 drugs are targeting only 549 molecular targets, which is 18% of the druggable genome. So we are still lacking drugs for 2,500 targets. So there is still a lot to discover and a lot to do. So 82% of our druggable genome is still uncovered, is still not attacked by small molecules. So there's still a lot of work to do and a lot of targets to discover. Okay, but do we really need small molecule chemistry, or can biologicals replace small molecule medicinal chemistry? Well, there are some arguments for, but only superficial arguments. If you look closer, these are no real arguments. Let's go to the superficial arguments. In 2016, 8 out of the 10 top global best-selling drugs were biologics. So you may think, okay, forget small molecules, invest in biologics. But if you not go for the selling for the price, but you go for the daily dose, then 0 out of 10 is a biologics. Why? Biologicals are very, very expensive, so they cost a lot of money, but on a daily dose, they are not too much spread out and too much important. Humira is adalimumab. Is still expected to be the top selling drug even in 2 years with $15 billion and will become the biggest blockbuster of all time for sure. The checkpoint inhibitors are expected to become the backbone of cancer treatment, also no small molecules, and even CAR T-cell therapy will make a contribution to cancer treatment. All of this are no small molecules. So do we really still need small molecules? Yes, we do, because the breakthrough therapeutic innovation is still coming to 2/3 from small molecules and only from 1/3 from biologics. And if you look to the new targets you approach in phase 1, phase 2, also about 2/3 are coming from small molecules and are not coming from biologicals. So we need definitely both, and there is still a major contribution of small molecule medicinal chemistry to very recent new drugs. An example, you know adalimumab, the monoclonal antibody against TNF-alpha, which is the top-selling drug now worldwide. And we have now a small molecule, so-called JAK inhibitor in the same indication, rheumatoid arthritis. And look what you can see here, 4 milligram of this tiny cozy molecule do exactly the same than this adalimumab, which is an expensive and very difficult to make as a biological drug. So a small molecule can do the same. A small molecule can really replace a very difficult, expensive, and hard to get biological. This is just one example from the field I know, from the kinase field. But let's look a bit back. In the last 20 years, we had very, very major breakthroughs in 3 areas: in HIV, in hepatitis C, and in cancer. And the really important breakthroughs in all the 3 fields came from small molecules. So HIV was not beaten by a vaccine. We still do not have a vaccine against HIV. It was beaten by the so-called HART therapy, by inhibitors of the HIV reverse transcriptase, integrase, and protease. So this more or less made HIV to a chronic disease, which you can survive for 20 or 30 years. 20 years ago, people died within 1 or 2 years. Hepatitis C, again, hepatitis C was not beaten by a vaccine. We still do not have a vaccine against hepatitis C, at least not a good one. It was beaten by sofosbuvir, which is a 20-year-old drug, but nicely packed in a prodrug. Even a more than 20-year-old molecule made as a prodrug is now the leading cash cow and the leading drug to treat, not only to treat, to cure hepatitis C. You can cure hepatitis C. 10 years ago, nobody would have thought that you can achieve this with a small molecule. And in cancer, the biggest improve in quality adjusted lifetime came from kinase inhibitors. And again, this is a small molecule and it's not a biological drug. So to make it clear, you need both. And small molecule chemistry here did the job, not vaccines as predicted 20 years ago. If you go to literature 20 years ago, HIV, hepatitis, and cancer was predicted to be beaten by a vaccine. And it's not the case. So there's still a lot to do, but on the other side, we have good chances to do so, and even from academia. So I will end now with this nice chart. This is me, and this is a medical doctor. And I'm telling the doctor here, I'm sure this stuff works, but you have to find the appropriate disease. Okay, this is more or less why you need both. You need a chemist, you need a doctor, and the two have to like each other, otherwise it would not work. And this is unfortunately now a phrase in German from a French Nobel laureate, from Albert Camus. I translate it to English. If you want to do something, you will find a way. If you don't want to do something, you will find an excuse. So what we have to do, let's look for a way and don't waste your time with excuses why it cannot work. Thank you.
Thank you so much, Stefan, for the very nice talk. We have time for 2-3 questions, but before that, I'd like to excuse ourselves because we didn't do your proper introduction to the audience. So to correct that, I would like to only to point some or highlight some achievement of Professor Stefan Walf, okay?
No, don't do that.
No? Really, because
No,
you have the
no.
audience, YouTube
No,
channel.
just introduce me as the old monkey, Macaco Velio.
So you are macaco velho that have a lot of achievements. So if you prefer not, okay. Are you sure?
It is perfect. I'm absolutely sure.
Okay. So we
So
have
give
time.
more time to Ricardo, the young people.
Okay.
Should
We have time for a question. The question can be right on the chat or do it by activate the microphone. Stefan, do you prefer that the question comes before or after Ricardo presentation?
It's up to you.
I prefer before because you can stop and go to the bed because I know it's almost midnight.
No. I'm curious to Ricardo's presentation. I've never heard this presentation, so you know.
So
I want to learn something as well. Huh?
Okay. So we will do the questions after Ricardo's presentation. Okay, Stefan?
Yes. Okay, I stop now my presentation. Okay.
Okay. Now I will do the introduction of Dr. Ricardo. It's possible?
Can I share my screen here?
Yes, Ricardo, please. We are seeing.
Oh, okay, can I see I'm just going to change the slide. Can you see the next slide just to see if it's moving properly?
It isn't in presentation mode.
Okay, then I think I'm going to do another way. Just a second.
No, you just have to start presentation mode.
Yeah, because I had this problem before, so I know how to solve it.
Right.
Now it's okay. No,
okay, now
not yet.
Can you see the
now?
next?
Yes.
Yes. Okay.
Yes.
Yes.
Okay.
Everything perfect.
Now we start the presentation of Dr. Ricardo Serafim. Okay, Ricardo?
Okay.
Dr. Ricardo Serafim studied pharmacy at Catholic University of Santos and received his PhD at the University of São Paulo, working with anti-decrease compounds. During his PhD, he had research stays at the University of Campinas, working with Professor Luiz Carlos Dias, and in the University of Pennsylvania, developing new methods in organic synthesis. In 2016, he joined the Structural Genomic Consortium at University of Campinas as a postdoc researcher in charge of establishing the Synthetic Medicinal Chemistry Lab. While being an SCG member, he did research stays at the University of North Carolina at Chapel Hill and at the University of Tübingen, where he is now working as a postdoc researcher. His main research interest is on the design and synthesis of reversible and irreversible kinase inhibitors, focused on generation of chemical probes to investigate function and therapeutic potential of unstudied kinase. Ricardo, the virtual floor, as we normally say here, is yours. Be welcome.
Thank you very much, Lydia, for a very kind introduction. And also Professor Elizabeth and also Professor Eliezer for the kind invitation. Thank you very much. Can you hear me properly and also see the slides, right?
Yes, perfect.
Okay, if during my presentation you have any kind of problem, please just, you can just interrupt me any time. Okay, I will also try to but the long story short, I'm just going to introduce you, I'm going to present you the development of the first covalent monopolar spindle kinase 1, MPS1 TTK inhibitor. That was our recent publication. And okay, but first of all, I just would like to introduce you a little bit more about protein kinase. And we know that the protein kinase are very important family of proteins that we have in the human body. And also we have more than 500 of protein kinase members, and they are all distributed here in this kind of phylogenetic tree that is called human kinome. So according to this structural similarity of these proteins, they are split here in different groups. And basically, just to have an idea of how important those proteins are, up to 30% of all human proteins, they may be modified by a kinase activity. So basically, most of the biochemical pathways that we have in our body, we are going to have several kinase acting. And just to have an idea, in this very normal biological event here in mitosis, so here we have several kinase acting, including one, including the most important one is the MPS1 that I'm going to talk to you in this presentation. Okay, but how the protein kinase, they can regulate other proteins and how they can modify other proteins. Basically, they can do that adding covalently adding a phosphate group to another protein that we call protein substrate. And this event is called phosphorylation. And as soon as some protein, the substrate protein, is phosphorylated, they can change the activity, they can change the localization in the cell, for example, or they can also change the interaction with other proteins. And this phosphate group for the phosphorylation comes from a molecule of ATP. So basically, a molecule of ATP will interact with the kinase, and then the kinase will transfer the phosphate group to the protein substrate. So if you look at the general and the representative structure here of the protein kinase, we can see that we have basically two lobes, the smaller N lobe and the bigger C lobe here. And in the middle, connecting both lobes here, we have, if you look here in the bottom scheme here, we have this part that I'm showing you. To here, I'm passing with my pointer, that is called hinge. This hinge region is the part that is connecting both lobes. And in the middle, between the both lobes here, we have the ATP binding pocket, as we are here well represented here in this part, in this figure up here. And basically, in this, if you could have a zoom in the ATP binding pocket, we're going to see the ATP here, the adenosine from the ATP interacting with the amino acids from the hinge. Basically, they are interacting, performing bihydrogen bonds here. We also have some other parts, for example, the sugar pocket, and also the region when the phosphate groups will be accommodated to be transferred to the protein substrate. Okay, and basically, since we have here the ATP are interacting by hydrogen bonds with the hinge region of the kinase, this is why when we look to some structures of the kinase inhibitors or kinase drugs, for example, specifically, we are basically going to see most of them with some heteroaromatic groups, especially heteroaromatic groups containing nitrogen, because this nitrogen atom can perform hydrogen bond with the hinge region. So, and here we can see different chemical structures, different scaffolds, but basically all of them can do the same. That is performing some, most of the time it can be 1, 2, or also 3 hydrogen bonds between the chemical scaffold and the hinge region of the kinase. They are basically mimicking the ATP. Okay, and another interesting data to see the importance of how fruitful the protein kinase inhibitors are for the treatment of diseases, in special cancer. You can see here that in 2020, we have 18 small molecules FDA approved in 1 year, only in 2020. And from this number here, from these 18 new molecular entities, small molecules, so 8 of these 18, they were kinase inhibitors. So almost half of the molecules approved for oncology field, they were kinase inhibitors. So just to see how strong is this field and how fruitful it is, especially in the oncology field. Okay, and then as Professor Laufer has already mentioned, so far we have more than 70 FDA approved small molecules being protein kinase inhibitors, and this number is continuously growing. And of course, in the next years, we are going to have a good improvement in this number as well. Okay, and also if you look here, this is a very recent publication, interesting, showing the compounds that we have in clinical trials. So basically, we have several molecules in clinical trials in phase 2, phase 1, and also phase 2 here. However, we still have many kinases that basically we are not, we don't have these proteins fully characterized, and also most of them, we don't have even good inhibitors for these compounds. Here, as you can see, 70% of the kinome is still considered unexplored. It means that there is no agent for which these kinases are considered primary targets that have entered in clinical trials. So I like this next figure that I'm going to show you. That's a kind interesting illustration about this number, that basically most of the people are taking the same road, and there are a lot of roads empty that has not been taken. So it means too many roads not taken. And if you are interested to learn and to read a bit more about the protein kinase, there are especially, and also about the understudied kinase that we call these proteins that are not being so far well characterized. I would recommend you here 3 interesting publications. These 2 publications here in the Nature Review Drug Discovery. Here, the first one is showing the kinase, how is the kinase field after 20 years of the imatinib, imatinib approval, and also this other publication here showing the trends in the kinase drug discovery. There are 2 very nice papers in this field, and I also strongly recommend you, if you are interested to understand a bit more about understudied kinase, in special how to design and how to create a very important and very powerful chemical tools to study this protein kinase. They're called chemical probes. I would recommend you here to take a look in our recent perspective article in the JMed Chem that we have published. Is quite interesting. I would like that you could give some attention on it. Okay. Now, specifically talk about the covalent inhibitors. Basically, some time ago, the covalent inhibitors, they are basically discovered by serendipity. But nowadays, we have a strategy, especially with the advent of the structure-based drug approach. We can design compounds and inhibitors and can act specifically in some amino acids from the targets that we want. Then basically these compounds can perform covalent interaction with specific molecular targets. And basically, as we know, many of the compounds, many of the drugs that we have in the clinical so far, for example, these 6 drugs that I'm showing to you here, all of them, they are covalent inhibitors. And if you look more specifically, all of them, since they are covalent inhibitors, they have, as we called, a warhead group. There is some reactive group that is going to perform a covalent interaction. And from these molecules here, I would like to highlight the afatinib, that was the first one, the first approved covalent kinase inhibitor. Okay, but when we are developing covalent inhibitors, we have, of course, some advantages, some disadvantages as well. So consider as some advantages, most of the time you're going to have a very high biochemical efficiency, and it means that also most of the time it may translate to lower dose and also reduce off-target effects. And this can be translated to a very good selectivity. And also with covalent inhibitors, we could uncouple the pharmacokinetic from the pharmacodynamic, and can have some prolonged duration of time, since the compound is going to bind most of the time in the irreversible covalent way. We can also design reversible covalent molecules as well, but most of the time we have an irreversible covalent interaction. And then the next, when if you are talking about drugs, for example, here, if you look, the next time that we have to make the administration of the compound, it's not going to depend anymore about the pharmacokinetic, but it's going to depend about the turnover of your protein, because since the molecule is bound to the kinase, basically, if it's an irreversible inhibitor, basically the protein is going to be bound to this compound, and the protein will be most of the time dead. And also, since we have a covalent inhibitor, we could also mitigate the development of some mutation in the binding site. Also, again, because we are going to reduce the administration, how often we are going to do the administration. And some disadvantage that we have for the covalent inhibitors, in some cases, we can have some problems of idiosyncratic toxicity and also some immune-mediated drug hypersensitivity. And if you don't control properly the reactivity of the warhead that we are using, this is why it's very important you know how reactive is your warhead and how is the behavior of this compound in that biological environment. You could have some problem, for example, hepatotoxicity, mutagenicity, and also carcinogenicity. And for some proteins that they have a very fast turnover, so the resynthesis of this protein is very fast. So in this case, you might not have a good advantage in this situation. But of course, depends of case by case of the protein that you are interested. And here, just some examples of the covalent protein kinase drugs. They are already in the clinicals. So as I said, afatinib, but we also have other ones, dacomitinib, neratinib, osimertinib, and so on. And most of them, as you can see here, except for the acalabrutinib, we have acrylamide as a warhead here, highlighted in red. But basically, we don't need necessarily to use only the acrylamide as a warhead. We have different other kinds of reactive groups that can be used as a warhead in the covalent design approach. But again, it's very important to understand the reactivity of this group and which place you are going to attach this warhead, the reactive part, in the reversible part of your molecule. And also, you have to adjust the angle and some other important properties. So all of these points, they're important to take a look in advance when you are designing covalent inhibitors. And basically, in special, when we have the acrylamide, for example, that is so far the most famous and common warhead, the mechanism that is performing the covalent interaction is by a microaddiction interaction here, but we can also modulate this interaction. For example, if you add the cyan group here, you can, instead of have an irreversible covalent inhibitor, you can generate reversible covalent inhibitor by that, for example. And first of all, when you have the covalent compound, it is going to interact with your protein of interest and the same time than a reversible inhibitor, only by reversible interactions. But by the end, since we have the warhead well installed and well attached in your molecule, it will attach covalently with some amino acids from your protein of interest. That most of the time, specifically in this case that I'm going to show you, is amino acids of cysteine. But we can also see in the literature some other case. We can also attach, for example, tyrosines and serines group as well. But the most common and the most frequent one are amino acids of cysteine. Okay, but when in the kinase field, I think the Professor Alpha will agree with me that basically it's not so difficult. You have you design very potent compounds for kinase, but the issue is to get the selectivity. When we are talking about kinase inhibitors, selectivity is a proper issue, especially nowadays. And here in this publication, in the Angevan, basically we have the distribution of all the cysteines that we have in the kinome. So this is called the cysteinome of the protein kinase. And every single here circle that you can see here, there are some protein kinase that has a cysteine in this position in the ATP binding site. And basically, I would like to highlight here this position here that you can see that is basically in the middle of the hinge group, is in a position that is called gatekeeper +2. The gatekeeper is this amino acid here. And if you go 2 amino acids further, we are going to find this position. It's called GK +2. And in this position here, in the entire kinome, only 5 protein kinase contains the protein, the cysteine in this position. So FGF4, the TTK, MPS1, the MAPCAP-K2, MAPCAP-K3, and the P7X6K beta. And I would like to emphasize here the TTK, also called as MPS1. That is the goal of my presentation today. Okay, this protein here, the monopolar spindle kinase 1, has a very important function during the cellular division, basically controlling the chromosome segregation. And we also have some studies have showed that the overexpression of this protein, for example, is now considered as a prognostic marker for neuroblastoma, for example. And there are also some other papers showing the importance of this protein, the overexpression of this protein in the development of some other kinds of cancers as well. And then basically, if we can inhibit this protein, we are basically going to have the mechanism. We are going to have the accumulation of the chromosome segregation errors, and then we are gonna have the cancer cell is gonna dead by apoptosis. Okay, and we have so far some reversible inhibitors already for the MPS1 that they are in clinical trials. For example, these 2 compounds here, compound 1 and compound 2 from Bayer, and also these other compounds here, compound 3 and compound 4 from other companies. But basically, all of them so far, they are reversible inhibitors. And also, if you look in the, when you take a look more in detail in the literature, we have realized that basically inhibitors with a long residence time might be more efficient to get the treatment for this cancer for the MPS1 inhibitors. It can treat this cancer more efficiently, and compare it with some compounds with faster dissociation kinetic. So, and this kind of property can fit quite well with the goal of the design covalent inhibitor for this kinase, because so far we didn't have any until the moment that we have published this paper, we didn't have any covalent inhibitor for this protein, only reversible inhibitors. And basically, as I said, in this region here, in the middle of the ring, the hinge, the MPS1 contains this cysteine here, the cysteine cis-04, and basically, as I said, only 4 other kinases contain the cysteine in this position. And also considering the importance of have a long residence time, increase the efficacy of the compounds against this protein, basically, we had as main goal of our project to have a proof of concept study for inhibiting the MPS1 protein covalently for the first time. And basically what we did, the first strategy that we did, we started to look at the PDB, the protein data bank, different kinds of, basically all kinds of reversible compounds that we have on the PDB to evaluate structurally if this compound we could, how is the binding mode of this structure, these different scaffolds, and if we could add some warhead and which position we could add this warhead in this scaffold. And basically, to make a long story short, we decided we decided to approach 4 different scaffolds. I didn't include the last one here because the last one basically it was chemically unstable, but we had worked on it as well. But basically, we synthesized a lot of compounds from these 3 different scaffolds here, in special from these 2 here, the scaffold C and scaffold B. But basically, we didn't have success in obtaining covalent inhibitors by this isoquinoline and this purine scaffold here. But fortunately, we were able to generate a covalent inhibitor using this 3-cycle scaffold here that synthetically was the basically the most difficult one, as I'm going to show you in a sequence. But okay, basically when we look at this reversible inhibitor here, this compound here from a group from Netherlands, they had this compound here, NETRC00660, that contains this methoxy group here. And as you can see here in this the co-crystal structure, we could see the methoxy group really close to the cysteine of interest. So they are basically close by 4.1 angstroms. That is considered a reliable value to add some warhead group here to perform a covalent interaction. So, and then we decide to include a warhead here, and the first approach that we used and so far was including the acrylamide as a warhead. Okay, and then we also decide to get to take a look a little bit close in this structural part, and then we decide to look at this other kinase here, the FGFR4, with this compound here, the blue 9931. That is, this compound here is the first covalent inhibitor for this kinase here, the FGFR4. And since this protein here also contains the cysteine in the same position, so we decided to make a overlay between the reversible TTK inhibitor and the FGFR4 inhibitor, and we could see here a good overlay between these 2 molecules. And then we realized, okay, if since they have a good overlay, if it's this strategy of adding acrylamide works for the blue 9931, it could also work for our compound against the MPS1. So, and this basically brought to us more confidence to keep working this strategy. Okay, and this was the strategy. We basically replaced the methoxy group here from the reversible inhibitor to acrylamide, the classical warhead acrylamide, to generate the covalent inhibitor called RMS-07. Okay, and just to show you briefly about the chemistry, as I told you, this was not a trivial chemistry, not a trivial synthetic route, especially talk about medicinal chemistry. And basically, just to have an idea, we need to use 7 synthetic steps just to get this key intermediate here. So it took a lot of time to get a good material of this guy here, this intermediate. And after that, with this intermediate in hand, we still had some other 7 steps to, by the end, obtaining here the desired compound with the acrylamide. And we also have synthesized here the compound, the RMS 191. There is the negative control, the non-reactive control. Then instead of the acrylamide, we have the saturated bond here. Okay, and then we first obtain the biochemical, perform the biochemical assays with this compound here. And just to have an idea how potent this compound is, we using the enzymatic assay, here we obtained the IC50 of 13 nanomolar. But this value here, we had to use approximately 15-fold the km of the ATP. Otherwise, we are going to have the limit of the assay. So we need to increase the amount of the ATP, and we can realize it here when we evaluate the Ki, that is the value independent of the ATP. And the Ki, we can see here the value of 0.83 nanomolar, so going to the picomolar range. And also what we realize a property of this compound that is a common property to the covalent inhibitors. There is, according to the time, we have the increasing of the activity here, as you can see in the IC50. So it was the first experiment that we did, that we started to realize that we could have a covalent inhibitor in our hands here. But it's always important when we are designing covalent inhibitors, it's always important to have our orthogonal assay to confirm the covalent interaction. And we also perform here the evaluation of the Ki, just to see how fast is this interaction between the warhead with the protein. And in our case here, this is, let's say, some weak point of our project, that so far we have a very slow inactivation kinetic. So the reaction half time was 69.8 minutes. Just to have an idea, some compounds, they could have this reaction half time of less than 10 minutes, or even less than 5 minutes, for example. So we are still working on it right now to increase and to make this inactivation kinetics higher and faster. Okay, and now we also perform some chromatogram. I forgot the name here. The mass spectrometry. Sorry, mass spectrometry assays here. We just purified the kinase domain of the MPS1, and here we basically add the compound for this for the isolated and the purified MPS1. And as we could see here in the mass spectrometry, we have here, this is the peak only of the protein, only of the MPS1, the kinase domain of the MPS1. And here you can see that according to the time after we add the compound here, the RMS07, we could see a mass shift of exactly the value of the compound. So it was a second assay that could confirm the covalent interaction being performed. And by the end, for the confirmation of the covalent interaction, we were luckily we were able to obtain the co-crystal structure that clearly we can see. If you go here in the right side, in the bottom here, we could see the electron density of the cysteine 604, the continuous electron density between the cysteine and our compound of this interest, clearly showing here the confirmation of the covalent interaction. And it was a very good resolution crystal with 2.0 angstroms. So basically, since we have confirmed the covalent binding mode, we decide to move further and also to evaluate other properties of our compound. For example, the target engagement and the selectivity, and also the initial preliminary data about the pharmacokinetics. Okay, and when we talk about target evaluation, target engagement, it's really important to know if the compounds there can really get into the target, the main target inside the cell. Because one thing is you evaluate the phenotype of your compound against the cell and compare with your biochemical assay, but you have to be sure that the effect that you are seeing in the cell is really because your compound is being able to interact with your protein inside the cell. So this is why we decided to perform a nanobreath assay. I will just briefly explain here this assay. Basically, we got our protein of interest, in our case, the MPS1, and we engineered, attached here a nanoluciferase, and we add the substrate, and also using a tracer that is basically a small molecule that can bind in the ATP binding pocket, attached with a linker, and also with the fluorophore, we have the tracer, and this tracer is able to bind in the ATP binding pocket. And as soon as this tracer is bound in the ATP binding pocket, and with the reaction of the substrate of the nanoluciferase with the luciferase, we're going to have bioluminescence resonance energy transfer here that we call as breath. And then we can get and measure this signal. And as long as you have a good inhibitor, this inhibitor will be able to displace the tracer. And then basically, if you have the interaction of your compound in the active binding site, and basically we're going to decrease the level of the breath signal. This is the main concept of the assay. And just to keep in mind that all of this experiment, all of this displacement, we are evaluating what is happening inside of the living cell. And this we can also have some ideas, not only about the target engagement properly, but also about some other properties, for example, cell penetrance, or also compound stability inside the cell. Okay, and then in this nanobreath assay here, we evaluated our compound here, the RMS-07, and we could see that this is also a very potent compound inside the cell with 22.4 nanomolar IC50 against the MPS1 in living cell. Another experiment that we did, since this compound is a covalent inhibitor, we also evaluated the residence time of this compound. And in this other graphic here, we incubated the compound for 2 hours, and then we evaluated here without any washout, and we can see basically no or a very low amount of the breath signal here. Again, when we perform the experiment again, but now performing the washout of the cells, we could see just a slightly increasement of the breath signal here. So it means that most likely that the breath signals are not able to increase because part of this protein is covalently attached to our inhibitor. As just for comparison, this AZ3146 compound here, when also we perform this experiment without the washout, we are going to see here also just a very low amount of breath signal. However, since this compound is a reversible inhibitor, after the washout, we are going to have basically the recovery of all the breath signal here. What is a common property in this case for reversible inhibitors? Okay, in the case of selectivity evaluation, the first thing that we did, we evaluated in a small panel of approximately 100 kinase, to be more precise, 97 purified human kinase plus 3 that are considered common off targets for kinase inhibitor, 3 bromodomains. And if you can see here, all of these 97 human protein kinase, they were spread in the whole kinome here. just to have a roughly idea about the selectivity against the entire kinome. And so in this small panel, basically we perform a DSF assay that is basically a thermal shift experiment, and we saw that we have only the MPS1 plus 7 other kinases that we have the value of more than 5 degrees. We are able to displace the protein more than 5 degrees. So what is quite interesting results for the first evaluation. And also another question that came to our mind, of course, and how is the selectivity of this compound against the other 4 kinases that contain the cysteine at the same position? And so when we evaluated that, we could see that the selectivity against the MPS1 is much higher compared with the other kinase. So basically we are in the micromolar range here for this other kinase, and here using the Km concentration of the ATP, and when we perform the assay in the biochemical assay against the MPS1, we are, as we have seen, we are in the basically in the picomolar range. And also, we just had a preliminary evaluation of the metabolism of this compound. First, we're evaluating the glutathione stability assay. That basically, because this is the most common nucleophilic compound that we could have in the cells, we have a high amount of this compound inside the cell, and basically we compare with the afatinib, that is already approval drug, the first covalent kinase approval drug, and we can see that the stability of our compound, the RMS-07, is even considerably higher than the afatinib. It means that basically we don't have any non-specific reaction with our compound. And also we decided to evaluate the stability of our compound in mouse liver microsomes here in an in vitro experiment, and we could see that we have a quite interesting preliminary result with the half-life of approximately 16 minutes. And we also were able to have an idea, we didn't identify it properly, but we also have an idea about one possible metabolite that we could have. That is, with this mass here is probably some hydroxylation. We don't know exactly which part of the molecule, but we are still looking at it as well. Okay, and by the end, we decide to check if this biochemical result could also translate to cellular results, and if we are able to treat cancer cells with our compound. And basically, we did like a very small screening in different kinds of cell lines in breast cancer cells, in colon cancer cells, and also in cells of hepatocarcinoma. And basically, all of them, we could see that our compound, the RMS-07, the covalent one, was quite active in a nanomolar range, in a middle to low nanomolar range. And also when we compare here with the non-reactive analog, with the saturated analog, the RMS-191, we could see that for all cell lines here, the covalent one was much more active compared with the saturated one. So which shows the importance of the covalent interaction to the efficacy of this compound. So I hope that I didn't go further with my time, but just to show it, basically by the end we were able to design and synthesize the first-in-class covalent MPS1 inhibitor that is called RMS-07, as we published this result in the JAMA Can some months ago. And also, I will not go in detail about this research, but also the PhD student here, this Stefan Gerstenecke, he also were able to design a covalent inhibitor against this other protein here. There is also one of the proteins that contain the cysteine in that position that is called ribosomal protein S6 kinase beta 2 in a very highly isoform selective way. And in this case here, he used SANAR warhead here instead of use with a microaddiction mechanism. Here is a quite different mechanism and a more recent kind of warhead. It's a very nice publication as well. And if you are working with the covalent inhibitors, if you are going to this field, or if you know someone that is working in this field, so you are highly welcome to submit some publication in our special issue of the journal Pharmaceuticals here, with a very interesting impact factor 5.8. That is, I'm sharing the guest editor here with the junior professor Matthias Geringer, and also with the Dr. Chiara Borsari. So the deadline is 15th of October of this year. And just my last slide, I would like to thank the research. This, I think I didn't mention in the beginning, this research was basically a collaboration between the SECMED of Unicamp, the Center of Medicinal Chemistry, and the University of Tübingen here. And I would like to thank in special here, the name of all the researchers, but in special from the SECMED, I would like to thank Andrea Santiago, that is also the first author, together with me, and also Rafael Cunhago from the SDC Frankfurt. They also have a very important collaboration in the selectivity assays and also in this nanobreath experiment, Martin Schwann, and also Professor Stefan Knapp. And here from the University of Tübingen, I really would like to thank Professor Stefan Laufer, and also the junior professor Matthias Geringer, that I'm currently a postdoc in his lab here. And then we can see here is the junior professor Matthias Geringer, is a very preeminent medicinal chemist here from Germany, and also all of his students and myself here. Thank you very much. I hope that I'm on time. And here, if you have any doubt or any question, you can just email me. And this is a very nice picture that I like to put in the end of my presentation from the Tübingen. Thank you very much.
Thank you, Ricardo, for the excellent talk. You have time for question. Very elegant work, Ricardo. It's impressive. Very well done. I have João Alberto. Would you like to make a question? Exactly, João.
Yes.
Please.
I'm sorry about the question before this presentation. Sorry. I have two curiosity about the first presentation to the Professor Stefan Laufer, and after that I would like to make a question for Ricardo also. If I could, I can.
Yes.
First for the Stefan. Professor, thank you for this presentation. I would like to know your opinion. Do you think that if to do more investment in project with lead optimization could be one good strategy for discovery of new drugs?
Could you repeat the question, please?
Do you think that if make more investment in academic project about the lead optimization, this could be one good strategy for discovery of new drugs?
Yes, sure. This is what I tried to show with these financial figures. In academia, we are not better in lead optimization, for sure not than industry, but we are cheaper and we can make it more parallel because our groups can easily make 5, 6, 7 PhD or postdocs in a project, and this is in industry, at least to the best of my knowledge, not the case. They have the lab with the lab head and 1 or 2 technicians, and they are working on the project. And as said, a lot of money is spent for this phase, and we can't do it cheaper, let's say it like that. And there is a big, let's say, shortage in medicinal chemistry in the developed world. In the last 10 to 20 years, everything was more or less exported to India or China, and there is a lack of competence in the meantime. And this can be filled up by academia much easier and much faster than it can be done by industry.
Thank you. The next question is, I have a curiosity about that you, Ricardo, said that the target in this case, all of kind of kinases. You said that we don't know all of these characteristics, for example, the binding site, but we know that we have some similarities about with these kinases. So I think that, what do you think when we discover all of these structures about these proteins? You think that we can see one larger change in the scaffold for discovery of new drugs, in this case inhibitor kinases? You think that we see the same scaffolds for these inhibitor kinases, or when we discover a new characteristics about these proteins, we can see a new scaffolds for this kind of and targets in this case? Can you understand?
Yeah, I think so. Basically, it's for me or Professor Laufer?
For you.
Okay.
It's for you.
So basically, actually, related to the structural part, we had quite a good advance during the last years, basically. But the main point is, as Professor Laufer has mentioned, is about the target validation. Because basically, most of the kinase, we don't know exactly what is going to happen if we inhibit this kinase, what is going to happen with the phenotype of the cell. Because if you are not using a proper tool for that, you cannot evaluate what is going to happen exactly. For example, let's say that I have a very promiscuous compound, for example, staurosporine, and then I just add the staurosporine that can inhibit, I don't know, basically the entire kinome, and then I'm going to see some phenotype. But this phenotype that I'm seeing is related to the inhibition of which kinase. You know, this is why it's important. We have that we call as chemical probes. That chemical probes have a very well defined properties related to the biochemical activity of this compound, with the selectivity as well. So we have the very well defined molecule that can inhibit a very few amount of kinase, or in a very good scenario, only one kinase. You know, and then since we have this compound, then we can evaluate the function of this kinase properly. So that's the main point. When I'm talking about the understudied kinase, it means that most of kinase we just have some of them. We have some genetic approach to evaluate, for example, with CRISPR, Cas9, and also with the RNAi, for example. But since we want to have a drug, the approach is different. We are not eliminating the entire protein, for example. We are just inhibiting the catalytic function, for example. And this we are only be able to study this protein since we have the proper tool for that. And this is why it's very important. And most of the kinase, they are understudied because we don't have a proper tools to study this kinase. This is one of the main reason for that. I don't know if I answer your question.
Yeah,
Okay,
yeah,
Ricardo.
but
And I don't know also if Professor Laufer want to add something in this topic.
thank you.
I don't
No,
think
you
so.
explained it perfectly.
Okay, Ricardo. I have a comment and two fast questions. The comment is, I think that the glutathione stability could be quite different if you perform the assay using a condition where glutathione transferase is present. Okay, because I have the impression that you perform the assay using a direct possibility of covalent reaction between glutathione and your compound, okay? In the absence of the enzyme. But the enzyme is very important to reproduce what happens in our liver. So it's only a suggestion that you look for to study the stability also in the presence of the enzyme. And
Yeah,
the question, okay,
yeah, definitely. Yeah, no, it's definitely, you're totally right. The point is that we just wanted to have an idea about how it could perform in a just chemically stability so far at this point. Because if we had some bad results only with the glutathione, like we probably would give up and go into another direction. But this was just something briefly that we want to understand in advance. But yeah, I totally agree with you.
Okay, thank you, Ricardo. And the question is, I would like to hear your opinion and Stefan's opinion about what you think about the possibility to use your compound to improve its activity or its pharmacotherapeutic profile, okay? Not increasing the structure, but reducing the structure. It means it's possible to make some molecular simplification and maintaining the activity, because I have the impression that looking for the bind sites, you have groups that are not doing any interaction with the enzyme. And the second question is, considering the particularity of the breath assay, is that possible that a compound that is interact with another bind site than the trace do, that is not be seen in your assay? Why? My doubt is, why you believe that all the compounds that you have prepared will compete with the trace bind mode? Bind site, perfect. Sorry, bind site.
This is basically like according with the X-ray structure, because with the X-ray structure, that for all these compounds that we performed this, how can I say, this analysis by the PDB, we got from the PDB, and we had this visual inspection, and we realized that all the compounds, they were binding the ATP binding site, and also we look at how we could attach the warhead and which position we could do that. So, and basically, as I said, we try different with some of the, for example, with some of purine compounds, and also especially the purine scaffold, we got a very potent compound from that scaffold. But and also we also had some X-ray from one of these compounds, and we could really see a very good electron density, but they are not continuously. So it means that we basically didn't have a covalent interaction. So as our focus were to obtain the first covalent inhibitor, basically decided to go to the other scaffold.
Okay, Ricardo, but let me clarify myself. The tracer, you show a slide teaching us how is working the breath assay, okay?
Yes.
And you put the figures showing that the tracer binds to the protein, and the compound will compete by the trace bind site. My question is, the trace bind site is the ATP bind site or is other one?
No, it's the ATP binding site.
Okay.
So basically, we bought it commercially. So we had all the information. But yes, it is the ATP. Would be really nice to have some assay that then we could, for example, to discover some allosteric inhibitor, for example,
Yes.
would be very
That's
interesting.
right. And the question about simplified compounds, Laufer and you also.
I would love it because it's gonna save a lot of time for me. Because especially right now that I'm basically scaling up in a multi-grams, like I think that I'm doing right now, I don't know, 10 or 20 grams scale. So I would love it. But yeah, basically, like, especially for the, just to comment that for the reversible part, that compound, the NTR, I don't remember the rest, with the methoxy group, they did a lot of structure activity relationship already with this molecule. So basically, they know they have a lot of background about the importance of the entire molecule. This is why right now we are especially focused in improve the covalent interaction.
Now, the problem with tiny molecules is not the activity. You can make even picomolar kinase inhibitors on a very small base, but you can never achieve selectivity with too small molecules. You need the interactions. You need probably 3.5, 4 interactions for picomolar activity, but then you need additional at least 2 to 3 to get the selectivity. So a small active and selective inhibitor is, for me, impossible, for other people probably hard to achieve.
Thank you, Stefan. Thank you, Ricardo. We have time for one more question or comment. No one? Really?
May I?
Oh, Professora Elizabeth, please.
Okay. First of all, I would like to thank you very much for the excellent presentation, and Professor Laufer for the excellent presentation in terms of inspiration in how to conduct our medicinal chemistry here in Brazil. And Ricardo, I compliment you for your excellent presentation and contribution to the cancer therapeutics with your compound. And I would like to know what is the further step you will present. Is there any industry interested in your compound, in your work? And this is not a matter of the consortium. I would like to know, what do you intend to do further?
To go further is the optimization of the inactivation property of the molecule. This is the main thing. What I'm doing right now, as I said, I'm really scaling up the compound, especially to obtain that key intermediate in a very large amount. Then I don't have to come back and synthesize this compound again. But yeah, that's the main thing right now, to optimize the inactivation property of this molecule, trying different warheads, for example, or something this related to that.
But is your intention to present the work to a further or a possible partner?
I think that not at this stage. No.
Right now. Okay, but in this stage, you can have a partnership, because there are some models that include pharmaceutical industries in the early steps of the research. So you wanted first to optimize, and after that to present to some possible partners.
Yeah, that's right. We have a lot of options later, but the first thing that I have to do is to really prove that I will be able to optimize it. But yeah, there are a lot of options after that.
And this is provided by the consortium, SGC.
No, actually,
They-
this the SGC somehow it's also involved because they perform, for example, the nanobreath assay and the selectivity. But basically, the main core of the idea comes from the junior professor Matthias Geringer here from the University of Tübingen. And then since I came here for the first time, 2019, then I basically started to move further with this project. But basically, this is a project, a collaboration between the main guys of this project, like here the Tübingen and also the SECMED from Unicamp.
Okay, so thank you once again very much, Professor Laufer and Ricardo, for your excellent presentation and a nice inspiration for all of us.
Okay, before we give back the word to the coordination for Professor Eliezer, I'd like to ask Professor Stefan what you can do to make part of Tübingen Academic Drug Discovery or to collaborate with Tübingen Academic Drug Discovery.
You know, we are already collaborating. So feel free to approach me with any of your problems. Feel free to send us PhD students in for sandwich or whatever. I think exchanging people is the best way of collaboration, you know.
Thank you. Some students is smiling now. So I think they are very happy to listen your answer to me. Professor Eliezer, please.
So disadvantage of Tübingen is that half a year it's cold, but advantage is that the beer is good. So Ricardo can comment on this as well.
Sure,
Yeah.
good beers,
Bad weather, but good beer.
good and cheap beers. I totally agree.
I fully agree with you. Okay, just to finish, I would,
Eliezer, just a moment. Someone is telling me that we have a question from YouTube. Stefan and Ricardo, are you agreeing to answer this question? Yes?
Yes.
Please, Ricardo, you can read the question on chat.
Is the chat on YouTube?
No, here. We copy to here.
Okay, because I'm not
The
seeing.
p38 MAP kinase apparently play a pivotal role in inflammation, but there are limitations as I target, I think. Limitation concerning selective blockade of p38. I mean, a toxicity seems to be big trouble here. I'd like to hear from you how druggable is p38.
No, this I can easily answer. There is no toxicity with blocking p38. The toxicity with the p38 inhibitors is coming from chemical toxicity, but not from mechanistic toxicity. You can make a knockout mouse for p38 and the animal is perfectly healthy. So there is no target-related toxicity on p38 alpha. There is one for gamma.
Perfect, Stefan. Now, Eliezer, please.
okay, to finish the session, I would like to thank you again, Stefan and Ricardo, for your very nice presentations. As Professor Ferreira said, in fact, the Laufer talk was very stimulating to academic work, and as he mentioned, the INCT-INOFAR is engaged in this process of drug discovery, and we are very proud to have Stefan as one of our external advisors. Thank you very much for your presentation. Also, Ricardo, with a lot of chemistry and a very nice piece of work. Congratulations to everyone to be here with us. And thank you very much for joining us, Stefan and Ricardo.
Thank you
Very
for
nice.
inviting us. Thank you.
Thank you
Okay.
too.
Okay, happy Easter holiday to everyone. And please, if you can turn the camera on, you can take a picture of everybody, okay? And after that, you can say goodbye to everyone.
Okay.
Hi, Lucas.
That's okay. It's enough. Thank you again, and bye-bye.
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 Thank you.
Thank you.
Bye.
you.
Bye-bye.
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 Bye.